Hello, aspirants, and a very warm welcome to this special explainer session from Rungta IAS Academy. Today, we are dissecting a highly probable, absolutely crucial topic for your UPSC Civil Services General Studies Paper 3, right under the Science and Technology module. We're looking at a major, real game-changer in cancer treatment, the height receptor.
Thanks for that introduction. So aspirants, to make sure this perfectly mirrors your ideal mains answer framework, our agenda today is straightforward. We'll cover the context and the primary hurdle, decode this concept of pseudo-heterogeneity, dive into the hit receptor solution, evaluate the Goldilocks challenge, and finally wrap up with the way forward for your personality test. Let's get right into it.
All right, let's dive into Section 1, GS3 context, and the hurdle, why CAR-T stumbled. Because, to understand the solution, we've got to clearly understand the massive problem facing modern oncology first.
Exactly. And for your introduction in a mains answer, you need a crisp, precise definition. So CAR-T cell therapy, or chimeric antigen receptor T-cell therapy, is essentially this revolutionary approach where a patient's own immune T-cells are modified in a lab. They're literally engineered to specifically hunt down and attack cancer cells.
Right, these engineered T-cells are isolated and equipped to mount a highly targeted attack. Now this therapy, it has completely transformed the treatment landscape for blood cancers. We are talking about leukemia and lymphoma. It has been a phenomenal success story.
But here is the catch, and UPSC examiners love a good comparative analysis. While CAR-T thrives against those blood cancers, it has severely struggled when applied to solid tumors, things like kidney, pancreatic, or ovarian cancer. The engineered cells simply could not eradicate the tumor completely, which allowed the cancer to eventually just grow back. We really have to ask, why the discrepancy?
Which brings us to the root cause of this failure. Section 2, Decoding Pseudo-Heterogeneity, the Illusion. This whole concept is based on some truly groundbreaking research out of Columbia University and the Sloan Kettering Cancer Center.
Okay, pay close attention to these steps for your notes. The researchers focused on a target protein called CD70, and they discovered an enzyme called EZH2. Let me repeat that, EZH2. What this enzyme does is it chemically modifies the proteins right around the cell's DNA. This causes the DNA structure to tighten up, which effectively drops the production of that CD70 protein to extremely low levels. Because the protein is dimmed, the cancer cell successfully hides from the CAR-T immune therapies. It's almost like an invisibility cloak.
This creates what we call drug-tolerant persisters. As cancer biologist Scott Lowe eloquently put it, these cells face intense evolutionary pressure. You see, they don't eliminate the target protein completely because they actually need a tiny bit of it to survive. Instead, they just hide it. The researchers termed this pseudo-heterogeneity, meaning the target is there, it's just way too faint to be detected.
So once this problem of pseudo-heterogeneity was identified, the scientific community desperately needed a targeted fix. Let's move on to Section 3, the hit receptor solution, bypassing the barrier.
And this is the absolute core of the breakthrough, the HLA-independent T-cell receptor, or the hit receptor. Examiners will actively look for this specific terminology in your papers. Instead of relying on a massive signal to trigger an attack, the hit receptor completely bypasses the traditional HLA genetic ID system. It just co-opts the T-cell's natural machinery, allowing it to detect antigens at drastically lower densities. It can sense even the faintest of signals.
And the real-world application here is highly encouraging. When tested in xenograft models, which, for context, use real human tumor tissues implanted in mice, the T-cells equipped with this hit receptor successfully eliminated those hidden drug-tolerant cells. They achieved a complete and lasting eradication of tumors in models representing kidney, ovarian, and pancreatic cancers.
Now, in Science and Tech Answers, you must always present a balanced view and discuss the challenges. So let's evaluate this in Section 4, the Goldilocks challenge, evaluating risks.
Precisely. We call this the Goldilocks challenge of cell therapy. It's a very delicate balance. Because think about it. If you engineer an immune cell with immensely increased sensitivity to detect those faint cancer signals, you inevitably run the safety risk of that exact same cell attacking perfectly normal, healthy human cells that might naturally carry minuscule amounts of that same CD70 protein.
Fortunately, to evaluate this risk, researchers mapped out a massive single-cell atlas. And the findings? Highly reassuring. Activity of CD70 was largely absent from vital organs like the heart, lungs, and brain. And while a small fraction, typically less than 5% of activated immune cells do express CD70 naturally,
The experimental models showed that any unintended attacks on these cells were temporary and, most importantly, medically manageable. Which brings us to our final segment, Section 5, Interview Focus, Way Forward. While you now have all the data for your mains paper, how should you articulate this during the UPSC personality test? An administrator must always think about safeguards.
So, if an interview panel looks at you and asks, how do we responsibly balance this extreme therapeutic power with patient safety as we move these advanced therapies into human clinical trials, what would your administrative mindset suggest?
Well, your answer should heavily focus on clinical prudence. The definitive way forward is the integration of advanced safety systems, specifically molecular switches. This ensures that if the therapy triggers an adverse immune response in a patient, doctors retain the ultimate control to simply turn the engineered cells off. It provides a necessary built-in administrative and medical safety net.
So, the crucial point we want to leave you with is this. As noted by geneticist and science communicator Anirban Mukhopadhyaya, this strategy is tackling the absolute biggest hurdle in cancer immunotherapy today. For patients with very limited options, uncovering these hidden cells makes all the difference. We leave you with this final thought to ponder for your exams. Will mastering the hunt for these drug-tolerant persisters finally be the definitive key we need to eradicate cancer regrowth entirely? Thank you so much for joining this explainer, aspirants. Keep studying and stay focused.